Good morning, everybody, and welcome to the next in our Connect on Coronavirus webinars from the British Society for Immunology. I'm Doug Brown, I'm the Chief Exec at the British Society for Immunology, and thank you very much for joining us because, again, I'm looking at the numbers here, we have hundreds of you who have signed up and are watching live, which is absolutely brilliant. It's great to see the momentum behind these webinars and the interest in all different aspects of coronaviruses and COVID. We've got more to come, so do keep an eye out on our Twitter and on our website and on our Facebook pages and the emails that we send out. So there'll be a lot more science coming your way over the next few weeks and months. And thank you also to the British Society for Immunology's journals, Immunology and Clinical and Experimental Immunology, for their support in making these webinars possible. As I've said previously, we're doing a lot in the coronavirus space, so please do, if you haven't clicked on it recently, have a look at our Connect on Coronavirus Hub webpage on our website. Lots of resources on there to help you, hopefully help you during these challenging times, as well as the recordings of all of the other webinars that have happened. So if you or your colleagues haven't had a chance to see some of these webinars live, all of the recordings are on our website, free to access to everyone, not just to BSI members. So please do spread the word and please do click and view those webinars. But for today, it's my absolute pleasure to introduce our two speakers. So we have two speakers today. We have Helena Maier from the Pirbright Institute and Erica Bickerton from the Pirbright Institute as well. They've told me they're not immunologists, which is absolutely fine. If anything, that makes it better. But they are here to tell us what we can learn about animal coronaviruses, which will be an absolutely fascinating presentation, and I'm sure a lot for us all to take away from the immunological perspective. So we'll have Helena speak first, and then we will hand over to Erica, and they will speak for around about 30 minutes with the opportunity for a Q&A session at the end for 10 or 15 minutes. So again, please do submit your questions via the question section on the webinar platform. So I will leave it there, and it's time to hand over to our speakers. And first up is Helena. And so Helena, I will hand the controls over to you.
Okay. So good morning. I am Helena Maier, and together with Erica Bickerton, we're going to present this webinar on what we can learn from other coronaviruses to inform us about SARS-CoV-2. Erica and I are both group leaders at the Pirbright Institute, specialising in coronaviruses, specifically coronavirus replication, virus-host cell interaction, pathogenicity, and vaccine development. And that's predominantly in livestock coronaviruses. When SARS-CoV-2 emerged towards the end of last year and developed into this pandemic that we're experiencing at the moment, it became obvious that there are lots of questions around the virus biology that we need to understand. So replication, how it transmits, epidemiology, evolution, and how we can tackle it using vaccines and antivirals. And there's an awful lot of research that's been done in these sorts of areas for other coronaviruses over the past few decades. So we're going to give you a bit of an introduction into coronavirus evolution, the replication cycle, disease, and vaccine strategies that are used for other coronaviruses. So coronaviruses are positive strand RNA viruses. I don't know if you can actually see that there now. They infect a diverse range of species. It's often thought that the humans and livestock viruses are the most important, and that's due to the economic and health impacts caused by infection. It's also been long documented that coronaviruses have an ability to cross the species barrier. And again, when this occurs into humans and livestock, that can have really big health and economic impacts, as is evident from the current outbreak. In addition to the current SARS-2 outbreak, coronaviruses really do continue to be a really important threat due to the very large potential for further incursion of novel coronaviruses into humans and other species. The coronavirus family is split into 4 different genera, the alpha, beta, gamma, and delta coronaviruses. And the ancestral viruses are thought to have circulated in bats and birds just through the diverse range of sequences of viruses that circulate in these species. The avian coronaviruses are then proposed to have given rise to the gamma and delta coronavirus genera, which predominantly infect avian species, whereas the bat coronaviruses are proposed to have given rise to the alpha and beta coronavirus genera, which infect predominantly mammalian species. Although other ancestral reservoirs are thought to also be important, for example, rodents and other small mammals, but these are perhaps underrepresented because there's less wildlife screening that's done in these species, so that will be really important to do in the future. Coronaviruses have 2 main routes of evolution. The first is through mutation of the genome during virus replication, and the second is through recombination of genetic material when 2 different viruses infect the same host cell at the same time. Unusually for an RNA virus, coronaviruses actually possess a proofreading activity. So the number of substitutions per position in the genome per year is actually up to 2 orders of magnitude lower than we might see for some other positive strand RNA viruses. But if we look at infectious bronchitis virus, a gamma coronavirus that infects chickens as an example, this virus has a really high R0 of almost 20 in unvaccinated naive populations. And if we put that into the context of a chicken house where there are numerous birds housed in close proximity to one another, there's ample opportunity for these viruses to replicate. This has resulted in numerous different serotypes and a really high number of strains that emerge fairly regularly. And this often results in antibody or immune response escape. And many of these viruses also have altered pathology. It was thought that this is a fairly unique feature of infectious bronchitis virus, although in reality, the screening for different serotypes and strains of other coronaviruses is not done to the same level. So it might be that we're just missing this for some coronaviruses. For example, the common cold human coronaviruses, we know that strains that circulate or have been circulating in recent years are not neutralized by antibodies that would target the reference strains from the 1960s. So clearly these viruses have evolved. Recombination is then the second route of evolution, and this tends to be important for switching host species. In an example here, turkey coronavirus emerged through recombination between related viruses and infectious bronchitis virus to now allow it to infect turkeys, causing enteric disease. And a similar situation is thought to have given rise to the pheasant, guinea fowl, and quail coronaviruses, although all of these viruses do fit into one single species. Cross-species transmission can also result in new viral species arising. So porcine delta coronavirus is an example here. The majority of delta coronaviruses circulate in avian species, although there are some examples that circulate in Asian carnivores, for example, the ferret badger and the leopard cat. Porcine delta coronavirus sequence was first identified in 2012, and it was linked to enteric disease in 2014. The virus is now known to have been circulating for much longer than that, and it's thought to have emerged in its current form in around 1993. The closest relative to other delta coronaviruses circulate in sparrows, and it's likely that the sparrow and porcine lineage diverged from one another at least 200 years ago, and this was probably through recombination of 2 sparrow viruses. There are some other really well-known examples of cross-species transmission, so we can look at SARS coronavirus and SARS-CoV-2. There are many SARS-related coronaviruses that circulate in bats, and some of these are able to infect human cells directly, although for the SARS-CoV outbreak, we know that there was an intermediate host of the civet, and the intermediate host for the SARS-CoV-2 outbreak currently remains unclear. MERS coronavirus was identified in 2012, and MERS-related viruses also circulate in bats, although it's worth noting that these are much more distantly related to the human MERS than the SARS-related viruses are to human SARS. Instead, MERS seems to circulate in camels, and seropositive camels have been identified throughout Africa and the Middle East. And it's now thought that there's fairly frequent transmission between camels and humans. So this provides ample opportunity for this virus to be able to adapt better to replicate and spread between humans. So this is another potential source for a future pandemic. Another example is the SARS coronavirus, which infects pigs. There was a large outbreak in 4 farms in China in 2016, resulting in a lot of deaths of pigs. and very closely related viruses were found to be circulating in bats when they were sampled from nearby bat caves in the preceding years. And significantly, this virus has also actually re-emerged in a farm in 2019, highlighting that there's the continued threat of these viruses crossing over, spilling over into a new species. And it's really important to continue surveillance and understanding of how these viruses, or which viruses, are likely to jump between species. We can also look at the evolution of the human common cold coronaviruses. So OC43 is a beta coronavirus. It's one of the major causes of coronavirus common cold. It fits within the beta coronavirus beta coronavirus 1 species, which is made up of a lot of bovine coronaviruses. Although you can see from here that although it's called bovine coronavirus, it infects a really wide range of species, and each of these isolates will be associated with particular genetic changes that make it better adapted to replicate within a given host. OC43 is thought to have emerged in around 1890 following transmission from cattle, and this coincides with reports of a respiratory pandemic at the time. All of these viruses are thought to have ancestral origins in rodents rather than in bats, and it's also worth noting that there is continued cross-species transmission between some of these viruses. For example, bovine coronavirus has been known to cause enteric disease in humans. Another human common cold coronavirus, 229E, which is an alpha coronavirus, has fairly well established origins in bats through camels. And if we look at the acquisition of mutations throughout the genome, mainly in the spike and one of the accessory proteins, we can see that there's this sequential acquisition of mutations from the bats through camels into humans. So despite existing in these 4 different genera, infecting different hosts, causing a range of different diseases, which Erica will talk about in the second part of this talk, there are commonalities amongst all of these coronaviruses about how they replicate, how they interact with their host cell, that can tell us quite a lot about novel viruses such as SARS-CoV-2 when they emerge. So I shall describe some of those now. The first stage in the virus replication cycle is attachment of the virus to the host cell, and as you'll all be aware, this is mediated via the spike protein. Spike is split into 2 separate domains, the S1 and the S2. The S2 predominantly is responsible for fusion of the viral envelope with the cellular membrane, whereas the S1 domain is responsible for attaching to the receptor. Within S1, there are 2 different regions that are involved in receptor binding, and these are used differentially between the different viruses, and also different viruses use different receptors, which you can see listed here. And indeed, some viruses actually just bind to sugars rather than to a specific protein receptor. Ultimately, interaction with the receptor results in fusion of the viral envelope with the cell membrane. However, the site that this occurs at, be it the plasma membrane or following endocytosis, varies between different viruses and even between different virus strains or different cells when you're studying this process. Ultimately, however, this results in release of the genomic RNA into the cytoplasm. Coronavirus genomic RNA has a 5' cap and also a 3' poly A tail, so it's directly recognized by the host cell translation machinery and is translated into 2 large polyproteins. These are encoded by the 5' or overlapping open reading frames in the 5' 2/3, of the genome, with a -1 ribosomal frameshift signal within NSP12. And this results in production of these 2 large polyproteins that you can see here. These are cleaved into the constituent proteins by protease domains within the polyprotein, and the enzymes for viral RNA synthesis, for capping, and also for proofreading are encoded within the 1B region. Most of these proteins have a pretty well conserved function between all coronaviruses. Expression of these proteins results in the rearrangement of host cell membranes to form what's called the replication organelle, and actually all positive strand RNA viruses do this. This replication organelle is thought to support viral RNA synthesis and also to protect viral RNA and synthesis intermediates from detection by the host cell, and that can be helpful, for example, in limiting type 1 interferon response. So the replication organelle for coronaviruses consists of double membrane vesicles, which you can see here and over here. And in addition, we showed a few years back now that in IBV infected cells, we see these smaller double membrane vesicles, which we've termed spherules. So they are pinched out from a region of the ER that becomes zippered together and they remain tethered. And these have subsequently been found in cells infected with coronaviruses from all 4 genera. So we can now say that the conserved replication organelle for coronaviruses is double membrane vesicles and double membrane spherules. Ultimately, production of the replication organelle allows viral RNA synthesis to take place. For positive strand RNA viruses, this occurs through a negative sense RNA intermediate, which is then copied back into a positive strand messenger RNA or genomic RNA. For coronaviruses, we get this 3' coterminal nested set of RNAs, which is shown here, with also a 5' coterminal region that's derived from the leader up here in the genome. This is produced during negative strand RNA synthesis. The polymerase copies along the RNA until it reaches this signal, which is called the transcription regulatory sequence. The polymerase can either read through this until it reaches the next TRS, or it can detach and reattach to a complementary sequence in the leader called the TRS leader. It then continues to copy along through to the 5' end of the genome, and this generates these shorter subgenomic RNAs. These are then copied back into positive sense RNAs, which act as messenger RNAs for the proteins encoded within the 3' end of the genome. So these are the structural proteins, which are the spike, envelope, membrane, and nucleocapsid, and these are all pretty well conserved between coronaviruses. And also the accessory proteins, which for this virus here is 3A, 3B, and 7. However, the number and position of accessory proteins varies a lot between different coronaviruses, and there's very little sequence homology unless they're very closely related viruses. Ultimately, expression of the full complement of viral proteins means that the virus is now able to regulate the host cell environment to create an environment that's favourable for replication. And obviously all viruses do this. They regulate a wide variety of different pathways, for example, the type 1 interferon response, stress responses, translation, metabolism, just to name a few. And the accessory proteins often play a really important role in this. So even though the number and sequence of the accessory proteins between coronaviruses varies quite a lot, we know that this is going to be a good starting point if we want to look at, for example, viral regulation of interferon signaling. However, some of the conserved proteins also play an important role. So just to give a single example, NSP15, which is encoded by the polyprotein, contains an endonuclease domain. Through mutation of this domain, it was shown to play an important role in limiting the type 1 interferon response by either degrading or shielding double-stranded RNA, which is associated with viral RNA synthesis. In the last couple of months, it's also actually been shown to cleave poly-U, which is present on the negative strand RNA intermediate, which acts itself as a PAMP. The final stage of the replication cycle is assembly and budding of or release of new virus particles. This occurs at the ERGIC. The membrane protein plays an important role in coordinating this and also in shaping the virus particle. M interacts with the nucleocapsid and spike to bring everything together, and RNA structures within the 5' end of the genome play a role in packaging. The envelope protein might play a role in pinching off the membrane to create the virion, although the precise mechanism of this is not really understood. Virus particles are released via exocytosis. This is not a lytic virus, so an infected cell will continue to release viral particles for several hours. So hopefully that's given you a broad overview of the virus replication cycle and has shown that although this is a new virus, we can actually already understand quite a lot about how SARS-CoV-2 interacts with its host cell and replicates itself, which will give us important pointers for how to perhaps develop antivirals that target conserved stages of this virus replication cycle. However, it's also worth noting that despite decades worth of research into coronaviruses, there are still a lot of unanswered questions. So it's really important to continue studying these sorts of aspects of virus replication for all coronaviruses. So at this stage, I will hand over to Erica to talk about coronavirus-induced disease and vaccination strategies.
Hello, thanks, Helena. So I'm going to talk to you today about some of the diseases caused by different host species. So some coronaviruses have been identified during screening programs, and their disease burden on the host is unknown, as is their potential for cross-species transmission. Whereas others are known to cause significant welfare problems or economic problems, particularly in livestock and companion animals. I'm going to just take you through a few examples of these coronavirus diseases that may have some relevance for COVID-19 in humans. So I'm going to start now with companion animals. If I can change the slide. Okay. So feline infectious peritonitis is caused by a rare in-host mutation of a common feline enteric coronavirus of cats. And this mutation changes its tropism from epithelial cells to macrophages. And this disease cannot be transmitted to other cats. And it often occurs several months to even years after the initial coronavirus infection. This causes a fatal multisystemic inflammatory disease caused by antigen-antibody complexes in the vessel walls, resulting in type 3 hypersensitivity reactions. And this disease can have one of two presentations. There's the dry form, which causes granulomatous lesions. These are in the thoracic and abdominal cavity of cats. Or there's the wet form, which is characterized by peritonitis and or pleuritis. And cats have vasculitis and increased vascular leakage. And there's been some reports that this may have some similarities to the Kawasaki-like syndrome that some children infected with SARS-CoV-2 experience. Another example from companion animals is the canine respiratory coronavirus. This is a highly contagious respiratory beta coronavirus that was first detected in 2003 in dogs housed at a UK rehoming centre. The spectrum of disease described for this virus ranges from asymptomatic through to kennel cough, which can be quite mild, all the way to severe bronchopneumonia. And the virus can be detected in a wide range of respiratory and lymphoid tissues, including the nasal mucosa, trachea, lung, and also occasionally in enteric samples. This replication in ciliated cells of the upper airways contributes to robust transmission of this virus. Now, there are a number of animals used for food production worldwide, and there's at least one coronavirus associated with each of these livestock species. Starting with camels, as Helena mentioned, these can be infected with MERS coronavirus, and this results in clinical signs similar to the common cold in young camels. Camels can be reinfected with the virus. But the key issues with this virus is that it can be transmitted to humans who may experience more severe respiratory disease. As Helena mentioned, bovine coronavirus infects a large variety of different ruminant species, so water buffalo, cows, goats, and sheep. This virus is a single serotype, but it has an unusually large host range for a coronavirus, and this isn't currently well understood. There are three distinct clinical syndromes that can be caused by bovine coronavirus, including diarrhea in young calves, winter dysentery with hemorrhagic diarrhea in lactating cows, and this coronavirus can cause respiratory infections of all ages of cattle, including the bovine respiratory disease complex and shipping fever pneumonia in feedlot cattle. Infection decreases milk production in dairy cows and can result in serious economic losses. An infection of calves can be fatal. There are no genetic markers that have been linked to any of these disease types or host range determinants. But there are vaccines commercially available against bovine coronavirus. The ones shown at the top there are inactivated virus that are administered intramuscularly, and these also incorporate other antigens from other pathogens. The idea is to vaccinate healthy pregnant cows in order to protect the calves. So the antibodies produced by the mother are fed to calves in the first few weeks of life to reduce the disease burden through passive immunity. And this protection persists as long as the calves are fed stored colostrum or allowed to feed naturally from the mother. So this shows that neutralizing antibodies can provide protective immunity through passive transfer along with ongoing mucosal administration. Pigs are quite unusual in that they can be infected with a large range of different coronaviruses. So coronaviruses of 3 different genera: alpha, beta, and delta coronaviruses. These viruses cause a whole spectrum of disease, from respiratory disease, diarrhea and vomiting, and wasting disease. And like bovine coronavirus, there's variable disease severity depending on the age of the animal and the immune status. There are new viruses emerging and new variants causing significant economic losses to the porcine industry. Helena touched on some of these earlier, but just to give you an example of this, when PEDV, porcine epidemic diarrhea virus, outbreak occurred in the United States a number of years ago now, the losses were estimated at more than 8 million pigs in the US, amounting to losses in the range of $900 million to $1.8 billion. So these really are significant diseases of livestock species. Just as an example of one of these coronavirus diseases, TGV, or transmissible gastroenteritis virus, was a major problem in the porcine industry for a number of years. This caused a fatal disease in young pigs, but also older pigs can be acutely infected in the upper respiratory tract. There are vaccines available, these are live attenuated vaccines, and they were licensed for use in healthy pregnant swine, again to prevent disease burden in piglets. These were either delivered intramuscularly or by oral prime, followed by an intramuscular boost around a week before farrowing. And again, like bovine coronavirus, protection is provided through passive immunity to the young. This was a case for a number of years. The vaccines weren't particularly effective, and TGV was still a major problem in the porcine industry until PRCV, or porcine respiratory coronavirus, emerged. This was first detected in the early 1980s. And this is a variant of TGV. It's evolved and it has a large deletion in the N-terminus of the spike gene. This results in a change of tropism of the virus from a mixed enteric and respiratory tropism to an almost exclusive respiratory tropism. This virus now causes upper and lower respiratory tract disease in pigs, but the infection is usually very mild or subclinical. However, the pathology in infected pigs is really reproducible, and infected pigs have lung lesions and pneumonia, even though they often experience mild disease. This means that its PRCV has been proposed as a model for SARS coronavirus infection of people. And after the emergence of SARS in the early 2000s, this PRCV model in pigs was used to investigate corticosteroid treatment of SARS infections. So PRCV is now endemic worldwide, and most pigs are now immune to both PRCV and TGV. And the emergence of this virus has significantly reduced the clinical and economic importance of TGV. And finally, I'm going to talk to you about avian coronavirus, or infectious bronchitis virus. This was actually the first identified coronavirus in 1936 to 37. It's the #1 cause of infectious disease-related loss to the UK poultry industry. And it's an economically important virus due to poor weight gain of infected birds and reduced egg production and egg quality. You can see here that infected birds produce poor quality eggs that have soft or misshapen shells. And this virus is endemic, although new strains and serotypes are constantly emerging, causing issues in the poultry industry. Vaccines are developed by taking a virulent field strain and then passing this through embryonated hens' eggs around 100 times to attenuate the virus. This molecular basis for attenuation is not currently well understood, so it's achieved by getting a balance between attenuating the virus while still maintaining immunogenicity and by a trial and error process. Because these are live vaccines, there's a risk that with a few changes they can revert to a more virulent phenotype. Some recent studies that we've done at Pirbright looking at 4 independent replicates that have been passaged in eggs of a virulent strain has resulted in different patterns of genomic variation in each replicate. In the attenuated viruses, there's very little overlap in the genetic changes, and the changes arise in the spike glycoprotein, so perhaps unsurprisingly, but also in the nucleocapsid protein and untranslated region at the 3' end of the genome. So these live attenuated vaccines are administered by the ocular nasal route, so by spraying the chicks in the hatchery or in the barns, or by putting the vaccine in the drinking water. These vaccines must be really inexpensive to produce due to the small profit margins in the poultry industry. And there's a number of issues, including the possibility of reversion to virulence, as I've mentioned, poor cross-protection between the many different circulating strains, and this amounts to a high risk of vaccine breakdown. This is a typical vaccination schedule for a layer chicken, so a chicken that produces eggs. So they're vaccinated multiple times, generally by spray with live attenuated vaccines, and then at the point before lay, they're boosted with an inactivated vaccine intramuscularly. And they're vaccinated with at least 4 different serotypes, including vaccination multiple times with the same serotype. And this schedule is designed to try and induce broader protection between the different serotypes. The breeders and layers are vaccinated to prevent egg production losses, as well as to pass protective maternal antibodies onto the progeny. And these antibodies are critical in providing protective immunity, although there's also a role for cell-mediated immunity that's not as well understood. So there's a number of new vaccine approaches for IBV that are being developed by us and a number of other groups. These typically involve using a vector to express the spike glycoprotein as a whole, or the S1 or S2 subunit, or by injecting recombinant protein. And these vectors include fowl adenovirus, fowlpox virus, Newcastle disease virus, and others. We've done some work expressing spike S1 or S2 subunits from a virulent strain in an attenuated IBV strain, and we found that immunising with a full-length spike from the virulent strain gives much better protection than the S1 or the S2 subunit, and that data can be shown here. We've also done some work into the cross-protective immunity of these vectored vaccines, and found that there seems to be little cross-protective immunity induced by expressing the spike in an attenuated IBV backbone in this way. So it's not clear yet whether we need better vaccine replication in target organs, or whether more than the spike glycoprotein itself is required for optimal protection and cross-protection. And none of these new IBV vaccine approaches are licensed yet. So just to summarise, control of animal coronaviruses is achieved by either controlling the outbreak or preventing outbreaks through vaccination. And there are vaccines available for several animal coronaviruses. But there are a number of problems with these, as I've described for IBV, but also problems due to high variability and poor cross-protection for new strains of virus, including PEDV and IBV, and also for emerging viruses such as the SARS coronavirus that Helena described earlier. So what can we learn from animal coronaviruses? It's clear that many different animal species are natural hosts for coronaviruses, and infection can result in different diseases and different disease severities, depending on the age and the immune status. And there's some commonalities between infection of animals and infection of humans with SARS coronavirus 2. There's some commercial coronavirus vaccines available. These are both live attenuated and inactivated. And it seems that prime and boost strategies can be effective. Maternal antibodies play an important role in protection of young in livestock species, but cell-mediated immunity is also important. But there are still many questions to be answered about animal coronaviruses and coronaviruses in general, around how they replicate and cause disease, and how we can best develop vaccines to control these. So it's really key that we continue to do research in this area. So I'd just like to say thanks for your attention, and we'd be happy to answer any questions that you may have.
That's fantastic, Erica and Helena. Helena, are we able to have you back on camera and on microphone?
Yep.
Excellent, good. Well, that was a fascinating presentation, and we've all learned a lot about coronaviruses, and lots of questions have come in, as you would expect. So I'm going to ask as many of these as possible. I'm trying to group them into different types of questions based on the amount of interest on different areas that's out there. So we're going to start with some questions around SARS-CoV-2 specifically. But the animal side of things. So you mentioned that the immediate host or intermediate host for SARS-CoV-2 is currently unclear. Do you have any inkling, and is there some emerging research that could shine a little bit of light on what that intermediate host could be?
So I think there's been a number of studies that have been carried out so far, but it's not yet clear what the immediate host is. So there's some evidence that cats can be infected and transmit to other cats. Dogs can be infected but less able to transmit. For example, chickens and ducks aren't able to transmit the disease. Transmission is and infection is a different question to which host species can be infected in cell culture, however, and there's mounting evidence that a number of different host species can be infected in cell culture. Obviously, more research needs to be carried out into the significance of this to real life.
And I mean, Helena, do you have any thoughts on that question?
I think Erica's probably covered that quite comprehensively.
Brilliant, thank you, Erica. And you mentioned that you know, cats and dogs can be infected. So we had a question that came in around that they can be infected, but is there transmission just between them, or is there transmission, ongoing transmission to humans and the owners as well? Is there some insights into what's happening in that situation?
I think there's a lot less evidence of transmission from animals to their owners. There's some experimental evidence that, as you said, cats can transmit the virus to other cats, but I'm not aware of a lot of research into whether humans can, the animals can transmit to humans. It doesn't seem to play a large part in the ongoing infections in humans, certainly.
Great, thank you. And actually, Helena, there's a question directed to you specifically, but I'm sure our viewers wouldn't mind hearing from you as well, Erica, if you've got some views. But the question is there evidence that the OC43 has become less virulent, and was the 19th century pandemic severe?
I can't say I know a huge amount about the 19th century pandemic, so how severe that was, I can't answer. In terms of whether it's become less of a problem in terms of disease in humans, I don't know that there's that much known about that. Erica, correct me if I'm wrong, but I think the common cold human coronaviruses have been fairly understudied, I would say, just because they cause a common cold. So kind of being able to get funding for that sort of thing is, or has historically been, a bit more difficult. So the majority of research into coronaviruses in the past has been into the livestock viruses, which cause obviously large economic problems, and then more recently into the pandemic, or the kind of novel coronaviruses that have emerged. I think it's a really interesting question, and I think doing much broader surveillance of circulating strains and comparing back to historical samples will be really important to gain this sort of information, which will inform the situation for the current outbreak.
That's great, thank you. So no, definitely that message of we need to understand more and we need to keep researching is coming across very loud and clear, and is one that the British Society for Immunology, of course, supports. So thinking about these other viruses, there was a couple of questions that came in about MERS, and specifically why you feel that SARS-CoV-2 has had more impact on global health and globally than MERS ever has. Are there some insights into the virology that could explain that, maybe?
Well, I think SARS, and particularly SARS-CoV-2, are just much better able to replicate and spread between hosts. So the mortality rate for MERS is pretty high, and you do get onward transmission, but it depends. It's usually in a kind of hospital setting or something like that. So I think the maximum has been about 4 steps of transmission on, whereas you're clearly getting vastly more than that for SARS-CoV-2. So what the reason behind that is not entirely clear. Clearly that it must be less well adapted to humans as a host compared to camels, where it will circulate. But the specifics, I think that's quite difficult to answer.
I bet, again, more research is needed. And so we had some questions as well, thinking that were referring to the different strains of SARS-CoV-2 and thinking about the impact on people with COVID infection. Is there, and are you seeing, from, again, that virology perspective, are the different strains having different effects in the body, respiratory versus kidney and others, or is that a more of a kind of host response rather than the virology?
I think there's very little evidence that there are different strains of SARS coronavirus 2 currently. For it to be a different strain, there needs to be an obvious difference in phenotype. And there's no evidence at the moment that the genetic variation that we're picking up during screening is resulting in a different strain. They may emerge in the future, but at the moment that evidence isn't there. So it's possible that the differences that people are experiencing in terms of disease severity and I think that may be down to the host rather than the virus. But again, it's actually really early in the life cycle of this virus, and there's a huge amount that we still need to learn about it.
That's great, thank you, Erica. And considering the time, and we did say we'd only keep you until 11:45, this is the last question, and it's based around vaccine and treatments, and you both referred to these in your talk. So it's clearly reassuring that a number of animal coronaviruses have effective vaccines. I mean, firstly, are you confident that we can develop, or how confident are you that we can develop a vaccine for people for SARS-CoV-2? And are there any specific learnings we can take from how vaccines have been developed in the animal for various coronaviruses? And, are we taking those learnings? Should we be taking more of those learnings for the human vaccine development? But also the antiviral side of things as well. And, do you feel that there's more that can be done to try and identify either existing treatments to repurpose or develop novel treatments for antiviral control of SARS-CoV-2?
So I think there's definitely more we can learn about antivirals, and there's clearly conserved mechanisms of replication between different coronaviruses. So we can use that information to test antivirals that are currently available, and hopefully identify new ones if we know more about how the virus replicates in different host cells and different host animals. And in terms of vaccines, yes, I'm personally hopeful that we will develop a vaccine for SARS coronavirus 2. And I think it's important to look at which animal vaccines are successful. Clearly, some are more successful than others, but the animal coronaviruses have slightly different challenges to developing vaccines in humans. But yeah, I'm certainly hopeful that it can be done for humans.
That is very reassuring. Thank you, Erica. Helena, any final words or thoughts on that question?
Yeah, I think I'm in agreement with Erica that I'm hopeful that it should be possible to get a vaccine that's effective. Which route that takes and kind of which kind of method of vaccination is going to be difficult. But for example, the SARS coronavirus in pigs, the second outbreak was managed through use of an activated virus vaccine. And that kind of vaccinating the farm meant that it didn't spread any further. So for novel viruses, then there is also precedent for being able to develop these vaccines. And in terms of antivirals, I mean, that's an area that's been researched. That's been a big focus of research for coronavirus researchers in the past years. So yes, I'm hopeful that there will be routes that we can tackle and prevent replication of this virus. It's just licensing things to go into humans is obviously needs to be very carefully regulated, so it just takes a bit of time, and it's not been that long since the virus was discovered. So you know, within the space of 6 months, then we've already come up with a lot of information.
That's brilliant. So I mean, on that positive, reassuring, but a well-measured response on how we will do potentially with vaccine development antivirals, we should draw this to a close. And I just want to say a huge thank you to you both. It was such a fascinating talk, and we can clearly learn a lot from what's happening in animal with regard to coronaviruses. So I'm sure you are telling your colleagues who do research in humans that that's the case. We will continue to do that as well at the BSI. So thank you very much for that wonderful talk, and thank you everyone for joining the webinar as well. As you know, we've recorded this and we will put this on our website, so if you want to see it again, by all means you can, but also tell your colleagues how amazing this presentation was, so that they can spread the word and more people can see these recordings online for free, for access to all. Also, don't forget the BSI is here to support you in everything that you're doing. So again, have a look at our website, but do get in touch with us if there's anything else you feel the British Society for Immunology can do to support you in your day-to-day work in the labs and in the clinic. So please do reach out to us. We want to do everything we can to support you at all times, but particularly during this challenging time. So I'm going to leave it there. Thank you again to the viewers for joining us. Thank you to our supporter journals, but a huge thanks to our two speakers today for, again, a wonderful presentation. So thank you very much, and we look forward to seeing you all next time. See you soon.
Thanks.